Yes.
Nothing. All right. So good evening, everyone. I want to hope and believe that most of you are trying to log in. For those that have already logged in, I think it is good that we save on time. And let me start by apologizing that we were unable to have this lecture in the morning because of some small miscommunication. I had prepared for the class, but then I think we were unable to share the link early. So you guys will bear with me as I run us through this lecture. I'm happy that you've already started a series of hemolytic anemia lectures, and this will just be a continuation of the various causes of hemolysis that may subsequently lead to anemia. I'll try to work within an hour. So those of you who may be distressed wherever you are, moving from one end to the other, you would bear with me. So my hope really is that we try to go through enzymopathies and membranopathies. And generally, these are just abnormalities associated with enzymes and membranes. We will look at some of the laboratory aspects that we could employ for diagnosis for the two disorders, and then possibly also have an overview on aspects of clinical presentation or the things that you may need to look for when a patient presents with a hemolytic process. I think, again, that may have already been presented from the previous lectures, so I'll not belabor so much on the clinical symptomatology. So my hope at the end of this lecture is that you should be able to describe aspects associated with enzymopathies and membranopathies, or some clinical components that could potentially make you think of an enzymopathy or a membranopathy. You should also be able to investigate and diagnose, and then see how to possibly manage some of the patients just in case. So as an overview, hemolytic anemia basically is an early destruction or a breakdown of red cells prior to their destined time or programmed time for death. And so there are various causes that have been associated with hemolysis. If you broadly clustered or grouped causes of hemolysis, we have immune-mediated and non-immune mediated. Among the immune-mediated may either be autoimmune, alloimmune, or drug-induced. Those tend to be immune-mediated, and then the non-immune, where the diseases of interest today fall. We have hereditary and acquired causes. Among the hereditary causes, if we all think about the red cell and look at the red cell from a structural perspective, there are three major issues in a red cell. You can look at the outer structure, which is the membrane. Remember, the red cell is anucleate. There is no nucleus in a red cell. And so for the red cell to survive, it requires enzymes. Those enzymes will be essential in sustaining the red cell. Critical in the red cell that occupies a lot of space, almost 2/3 of the space, is the hemoglobin. And so the red cell would potentially have challenges with any of these areas. And from a congenital perspective, any of these areas could actually be affected. And so you may manifest with a hemolytic picture as a result of any of these three areas getting affected. And I think we've given a special session to hemoglobinopathies, which shall not be discussed in this meeting. Can the person who is in charge of muting students possibly mute someone in the background? So the other type of non-immune mediated cause of hemolysis is infections or mechanical causes. You have a cell that is mechanically distressed, and you may have heard about Marcher's disease. That is a common phenomenon for soldiers who are marching, and sometimes that process of marching causes distress to cells as they pass through smaller vessels, and so hemolysis could be a possibility. So why this lecture today on the red cell membrane and enzyme disorders? I mean, why the discrimination between these two? The first thing I want to mention is that as scientists, we want to look at therapy. So there are questions around therapy. And the reason is because some aspects associated with either membrane or enzyme disorders can be prevented. And the prevention may even include avoiding certain food types or medications. The other reason is because some of these disorders have been associated with excessive enlargement of the spleen. And so patients will tend to present with splenectomy. And the question that we now have to maliga with is, do we remove the spleen to prevent the destruction of the red cells or not? So is splenectomy part of therapy for patients with this kind of disorder? And this has been a long discussion. Some people would prefer to do splenectomy as a mode of intervention, while others would just look at the spleen and try to look for other causes that may lead to hemolysis to prevent as a mechanism to salvage the spleen. The other one is genetic counseling overally. Remember, these are hereditary. Majority of these disorders tend to be hereditary. And so you want to look at genetic aspects and then just modalities of inheritance. Ladies and gentlemen, when you are dealing with the hereditary disorders, it tends to be very complicated because families get destroyed, families get broken down. You see a lot of blame games. And so, you want to ensure that appropriate counseling is done. If the disease can be passed on either autosomally or sexually, you also through the sex chromosomes, you also want to make sure you give appropriate information so that then couples can avoid scenarios that can lead to bad outcomes with these disorders. We also know that some enzyme disorders are associated with the X chromosome. And so if there is a damage or a mutation on the X chromosome, some of these diseases could easily be passed from one generation to another. And if it is sex-linked, then the question is which of the sexes is affected? Is it males? Is it females? How does it express and show itself? And all these are things that we need to consider. Majority of the membrane disorders, as we did mention, tend to be autosomal dominant, while enzyme disorders tend to be autosomal recessive. So if something is autosomal dominant, it means you just need one abnormality. And if it is recessive, then you need a double dose. So a mother and a father must give for the disease to manifest. It's also important for us to notice that we can better understand the pathophysiology of the disease when we understudy these two diseases, so that we can come up with therapies, especially genetic related, because gene therapy is now the norm to go for a lot of these hereditary disorders. A lot of companies are coming up with therapies that can manipulate the gene so that the gene can produce appropriate proteins or appropriate enzymes to correct disorders. And so studying membrane and enzyme disorders has become a key factor, medically speaking. So I'll shift gears very quickly and start talking about hereditary hemolytic anemia, the general features that we tend to see in these patients. And we know that anemia is a key factor. Patients will tend to have a reduced hemoglobin content. And so these are patients with head, fatigue, or tiredness, head, and dizziness and things of this kind. They easily get exerted, so they can't do quite a number of their routines. The other one, from a laboratory perspective, is they tend to see an elevation of a reticulocyte count. We know that the sine qua non of hemolysis is presence of the reticulocytes. And reticulocytes are young cells. So when cells are destroyed in whichever state, wherever in the body, then the body tries to compensate by producing younger cells. Those younger cells are the reticulocytes. They still have some nuclear, remnants. And so then will be picked as retics. And so reticulocytosis is a common finding in these patients. Because of the destruction of the red cell, you would also tend to find an increased bilirubin level. And the question might be, is it going to be direct bilirubin or indirect bilirubin? I think these are things you can start looking into so that then you can distinguish the various types of hemolysis. The enzyme that we have always associated with hemolytic anemias is the lactate dehydrogenase, LDH. And this enzyme obviously will be found in high levels. You'd also see the proteins that are essential in managing hemoglobin, like haptoglobin levels that will tend to be reduced. And then when you do a Coombs test, this would tend to be negative because this is not autoimmune related. The other general feature that we would tend to see in these hereditary hemolytic anemias includes chronic versus periodic hemolytic episodes. There are aspects that you will tend to see chronicity, and so the presentation tends to be continuous, chronic in nature, vis-a-vis periodic, where you have certain predisposing factors that then makes one to break cells. So the chronic versus periodic episodes may be something worth looking into. And so clinically, with certain findings, you can see pallor generally in these patients. You would see jaundice or icterus, and this is evident on the various mucous membranes. Because of breakdown of cells, sometimes you see these patients suffering from gallstones. And so when you start forming gallstones, obviously you look at the complications associated with the gallstones. I did mention that splenomegaly is a mega presentation, and so these are children that would have distended abdomens or patients with enlarged spleens, and so the abdomens, or abdomen may be distended, and then there may be dark urine, and of course stools may also change in color. So why do we talk heavily about hemolytic anemias, and more so on enzymes? So you would tend to realize that hemolysis generally occurs in different areas in the body, and if you look at the red cell breakdown cycle, this may occur extravascularly or intravascularly. You have hemorrhage, sorry, destruction, not hemorrhage. You have hemolysis that occurs within the red cell. On my extreme right, if you can see my cursor there, you have a red cell that undergoes changes within the vessel wall that gets destroyed. And so if you are to take blood from this patient, you can be able to tell the hemoglobin levels in circulation. And so they tend to have hemoglobinemia. The other one is if you look at the other products associated with hemoglobin, such as methemoglobinemia, there may be an increase. Then, of course, hemoglobinuria will be evident, and then hemosidenuria will also. On the other hand, if you have a breakdown of cells outside the blood vessel, which is extravascular, and this could occur in the reticuloendothelial system, you have the macrophages here getting involved, but basically overall you have a red cell that is destroyed, and so the components that would be released would either be the globin related, where you would end up with amino acids, or iron destroyed, and this may lead to iron binding into transferrin and subsequent storage in different parts of the organs, including the liver and other organs that may take up this ion. Or you basically have bilirubin generation that goes to the liver for conjugation or for poor conjugation, and subsequent release through the biliary system down to the intestines, and subsequently reabsorbed back for release through urine. So these are the two major mechanisms through which red cell destruction could easily occur. So having said that, then it is important for me to try and tie this up and see the role of the red cells, the red cell destruction and the role of the red cell in terms of the enzymes that may be essential in sustaining the red cell and the roles that they play. And I think it is important for us to understand that the enzymes in terms of red cell glucose metabolism is essential because you need energy to sustain the cell. And so for the body to sustain the cell, there are various mechanisms through which this glucose metabolism could occur. One is the Ebenden-Meyerhof glycolytic pathway, which I think you guys did ably while in Chiromo. And you looked at how glucose is broken down to lead to lactate formation. And some of the enzymes that would be essential in generation of ATP, including various products in between. So when you look at this particular pathway, the Ebenden-Meyerhof glycolytic pathway, you have some products like 1,3-diphosphoglycerate being converted to 3-p-glycerate using pyruvate kinase. The same pyruvate kinase is also essential in generation of pyruvate from phosphoenol pyruvate. So pyruvate as an enzyme is an essential component in this particular pathway. But when you also look at the pentose phosphate shunt pathway, you have another enzyme that is quite essential in conversion of glucose 6-phosphate to 6-phosphogluconate. And this particular enzyme is glucose 6-phosphate dehydrogenase, the G6PD. This same enzyme is also essential in ribulose 5-phosphate to fructose 6-phosphate. So it's an essential enzyme, key regulator in the pentose phosphate shunt pathway. And you'll also notice that in the process of, these metabolic processes, you end up with ATP generation, especially at the level where pyruvate kinase comes on board, and you end up with some ATP generation. And this can occur at two different phases in the process of glycolysis. So it is important for us to understand these pathways and the role of the enzymes. And there are so many other enzymes that we shall be talking about, but I think it is just important for us to realize that for all these processes to occur and for the cell to maintain its energetic requirements, you need these enzymes to play a key role. Having said that, we also have the Rappaport-Lou-Behring shunt pathway, where you have 1,3-diphosphoglycerate being converted to 2,3-DPG, and also the same 1,3-diphosphoglycerate to 3-phosphoglycerate. And the enzyme of interest here is a pyruvate kinase or a mutase. Sometimes we get a 2,3-biphosphoglycerate or diphosphoglycerate mutase, causing 3-phosphoglycerate formation. So you need enzymes in this particular step, and in the process, as I did mention, you end up with the conversion of ADP to ATP. So it is very critical, ladies and gentlemen, for us to be aware that these particular pathways where the body attempts to sustain its functionality, and just like any other cell, the red cell requires these enzymes basically for its sustainability. Now, so looking at the pathology around these enzymes, ladies and gentlemen, there are more than 40 enzymes that are involved in red cell metabolism. And I think I have mentioned some of the processes that are necessary for these enzymes to function. And as I did mention, you can look at other enzymes, and at the end of the day, you'll be able to appreciate more the various enzymes that are involved. What is of importance is that about 17 of these are linked with the clear cause-effect relationship. And I've given some examples here. And again, as I did mention, some of the enzymes are critical. So if you look at some of the enzymes that I've mentioned here, starting from D6PD, all the way down to basically, all these are the enzymes that, are, as I did mention, will function in the cell for different reasons. And as I had alluded to, you need to look more into all these particular enzymes. I'll pick a few as we move along with this lecture and talk about them. So looking at the enzymopathies and some of the clinical aspects, I want you to notice that the degree of the metabolic dysfunction, number one, depends on the importance the affected enzyme causes. If the enzyme is a rate-limiting enzyme, then you will have accumulation of the products associated with that particular enzyme. And so the clinical presentation will be heavily dependent on the accumulation of the product or the impact the end product would have ended with. So if the impact is a failure to generate ATP, you can imagine how the patient would present. The next other important clinical aspect that is of interest to all of us is the functional abnormality and stability of the mutation associated with the enzyme. If you know, the mutation is not as important, or the mutation does not have the level of stability associated with the functionality of that enzyme, then you may say, you know, milder effects. The other one is the ability of the body to compensate. So an enzyme could be affecting a specific outcome, but how does the body compensate to this particular expression of the enzyme? So most enzymes, as I did mention, are associated with extravascular hemolysis. You will notice that intravascular hemolytic processes, as I did mention, occur in the blood vessel. But when you look at these abnormalities associated with the enzymes, a lot of these tend to cause extravascular related hemolysis. And so the spleen and the other reticuloendothelial systems tend to be overburdened with this abnormality. So the clinical, you know, causes generally may be heterogeneous based on the level of compensation of the hemolysis. The other one is some of these clinical pathology can manifest as early as at the neonatal stage to tend to present late, either because you need a predisposer for them to manifest. You know, they have taken something that then causes hemolysis in these patients. Sometimes you might find an enzymopathy that has no clinical correlation between that deficiency and the clinical presentation of the clinical severity. And we'll be looking at some enzymes that tend to be exaggerated in terms of function. And so the clinical outcome may be completely different. While you are expecting a bad outcome, you find that possibly the enzyme is overstimulated. And so while it is a pathological overstimulation, the clinical presentation may be so different. While other, you know, abnormalities may be so severe and the clinical presentation does not correlate whatsoever with the role of the enzyme. We've also seen some associated with the non-hematological symptoms, like, for example, you see myopathy in pyruvate kinase deficiency and severe neuropathy in triphosphate isomerase deficiency. So you might actually see complete different clinical presentation involving different anatomic areas, yet the problem is actually an enzyme deficiency within the red cell. So again, the clinical presentation and pathology may be completely different. So ladies and gentlemen, I'll start by talking about G6PD, glucose-6-phosphate dehydrogenase deficiency. And I generally, you know, note to students that those of us who are highly mobile, you know, thank God COVID is limiting some of us from moving around. If you went towards the Mediterranean region, you know, this disease is said to be a big challenge. And unfortunately, being African, being Black, being in this continent has always been associated with enzymopathies, especially G6PD. I don't know how many of us have ever come across a patient with G6PD. I'm yet to see one myself. So anything bad generally is said to be in Africa. And I think this is the reason why, ladies and gentlemen, we present these presentations to you, so that we can change the course and the thinking around Africa. It's very unlikely that this disease is as common as it is sometimes mentioned. I may not give you any specific statistics, but this is what literature has, that the disease is common in Africans. I would believe, because I don't live in the Mediterranean region, but I assume that the disease may be commoner there. Just in case you find yourself getting married to a Mediterranean person, please watch over this particular disorder. This disease is said to be sex-linked, and so chromosome gets affected, and so patients would tend to pass this through the sex chromosome. In terms of classification of this particular enzyme, you will notice that you have class 1, where there is severe deficiency of this particular enzyme, and it is associated with chronic non-spherocytic hemolytic anemia. So spherocytes basically are small round cells with almost no area of central pallor, and this particular enzyme disorder, especially this class, tends to be non-spherocytic. But there is, for sure, there is hemolysis. Then the next one, which is class 2, has a deficiency of between 1 and 10. So class 1 generally is almost as good as being told there is nothing. It is less than 1%. So in class 2, you have some 1 to 10% residual activity of the enzyme, and you would tend to see an acute type of hemolysis. So the hemolysis that you would see tends to be acute, and so at least you have some residual activity of the enzyme. For class 3, this is moderate deficiency of between 60%. So there is something, and you may expect some hemolysis, but as I did mention, you might find that this may require some predisposition for one to manifest. Class 4 and 5, ladies and gentlemen, are always where the headache lies. These are normal individuals. Class 5 even has an increased activity. Someone might ask, so what happens? And it would be nice for us to now go and look at literature. What happens when you have an excess increase in G6PD in terms of activity? What happens? And patients would tend to have this kind of deficiency, but at the same time, look normal. The prevalence of G6PD is 1 in 10,000, and so it's supposed to be much commoner than we would think. In Kenya, we have a population of about 50 to 55 million. I think we are now at around 50 million. And so we should be having at least 5,000 patients with G6PD. This is almost synonymous to hemophilia and other bleeding disorders, where you have a similar prevalence rate. It's only that for G6PD, the distribution is a lot different as compared to other disorders. It is not associated clinically in terms of presentation with non-hematological symptoms. So a lot of its symptoms are hematologically associated. So in terms of hemolysis, you tend to have acute hemolysis, and this tends to be a critical phase in terms of presentation. So you have, hemolysis that causes some level of crisis. You can also get chronic or sporadic aspects of hemolysis. Now, G6PD is said to have some protective effect against malaria. Why is this the case? It is believed that if a cell that is G6PD deficient, on being infected by the malaria parasite, would easily be earmarked for destruction. Remember, it is already defective, and so we would assume that any malaria parasite in it is easily destroyed. And so G6PD has been said to have a protective effect. And where is malaria in the world? It is always an African problem. There is so much malaria in India, so I don't know how much G6PD is there. But anyway, that aside, there's also a high genetic polymorphism with triggers of hemolysis. And I think of importance really is to emphasize the fact that there are over 200 drug substances that have been associated with hemolysis in these patients, including what we give to manage malaria, primaquine. Okay, we have sulfonamides, nitrofurantoin, that may be associated with the urinary tract and the likes, phenacetin, etc. So there are many drugs that have been associated with a hemolytic outcome in G6PD. And so a patient who is on treatment ending up with some level of icterus or yellowness or jaundice as a result of medication sometimes may not just be an indication of hemolysis, but we need to suspect a G6PD. Infections can also cause a hemolytic picture and viral infections, and then patients will tend to end up with metabolic acidosis or jaundice, especially in the neonates, and even with to vitamin K. Now, there are foodstuffs that have been associated with the G6PD deficiency in terms of clinical presentation, and this includes Vivia fava. These are beans, and so favism is a key issue with severe hemolysis and hemoglobinuria. So there are products or food substances that have been associated with a hemolytic picture in G6PD. And so that is the molecular weight, and you can actually use different mechanisms. You can use sequencing to determine where the problem is, or you can run an electrophoretogram to look at the various variants of the G6PD, of G6 glucose 6-phosphate dehydrogenase as an enzyme, G6PD. And so these are just mechanisms that you could use. G6PD, ladies and gentlemen, is also said to be the most common normal type, historically speaking, of enzyme disorders. Okay, it's also a fast-moving non-deficient variant that is common in the Africans. They say it's fast-moving non-deficient variant and has no clinical significance. And I guess this is the reason why I initially teased us a little bit by asking how many of us have seen a patient with G6PD. And so with this particular caveat, it tells us that G6PD could be just as common, but then clinically speaking, it does not necessarily present as a hemolytic disorder. So some of us may be seated wherever we are with G6PD deficiency. Okay. There are over 400 variants of G6PD. Now, so there are different ways of looking at these variants. You have B, A negative, based on geographical and other trivial names that have been given to this G6PD, G6PD as an enzyme. As I did mention, the high incidence, of course, will be seen in malaria endemic areas, and it is thought to confer a selective protection against, especially Plasmodium falciparum malaria. Okay. I don't want to belabor on this because geographically speaking, as I did mention, G6PD deficiency is the most common metabolic disorder of the red cell with over 200 million people. So majority of the people and a large population would have this disease, especially in the tropical and subtropical areas. Okay. And we have tended to see G6PD as people start moving around in the world. And, the world is a mobile world. And you can all see that even as COVID moves around, that people move around. So as people move around, the disease also moves around. I will not belabor so much on the movement of the disease from one region to another. Okay. I'll not again belabor on this slide because you may not see Kenya here, but at least you can see an African country called Nigeria, okay, and the variant that is common in Nigeria. So you will see other places, exhibiting G6PD, and this is the frequency in males. As I did mention, we need to understand how the disease is moved from one client to the other. These are just presentations associated with the hemolytic picture. And as I did mention, you get an acute phase and also a chronic phase, but in an acute phase, all these are the presentations that you would see. And without belaboring so much, anything that has to do with the hemolysis, including abdominal pain, fever, hypotension, jaundice, renal failure can occur. And you can also have a recovery phase, which is gradual but rapid, and urine clears in fewer days, with the jaundice clearing within 1 to 2 weeks. So all these are things that we need to look into. Then here you have a list again of all the drugs, or some of the drugs that have been associated with the G6PD. I'll share these slides with you, and you can take your time to understudy these particular drugs. But of importance for me really to mention is that even drugs used to treat malaria have been associated with hemolysis. And so while we are treating malaria and we are thinking it is malaria that is worsening the jaundice, it could just be, again, G6PD. Okay. Other enzymes, and I would want you to go and study more about them, is pyruvate kinase, and we saw where pyruvate kinase was essential. It's a rare disease, but commonest enzyme deficiency in Europe. It's autosomal recessive with a prevalence of 1 in every 20,000. So we should be having at least 2,500 patients in our population that may be having this disease. And the causes for hemolysis tends to be severe to mild. So again, the disease may not be as common in our setup. And so I would encourage students to read on other enzymes just to understand them, because we only give you a guide on the commoner types. Let me then change gears, ladies and gentlemen, and start talking a little more on membrane-related disorders. As we all know, the red cell membrane is basically a skeleton that harbors the various contents within the cell. And so as a skeleton, we know that it is a pseudo-hexagonal meshwork of spectrin, actin, and ankyrin, with actin-associated proteins. So basically, we are dealing with proteins that form a mesh, basically that assists, support the skeleton and the functioning of the cell. So these particular proteins and some laminate the inner membrane surface and attaches to the overlaying bilipid layer. And these proteins are anchored by a band 3 that contains a multi-protein complex at the ankyrin and actin binding ends of spectrin. So basically, we are talking about proteins at various levels. Now, the membrane skeleton then, gets strengthened or strengthens the bilipid layer, and basically gives the membrane durability and flexibility so that then it can move from one end to the other. Remember, ladies and gentlemen, the red cell has to move within circulation from one corner of the body to the other. And sometimes it has to pass through very thin blood vessels. If you look at the capillaries or the venules, especially the capillaries, they tend to be very small. And so the cell has to change shape for it to pass through this particular lumen. And so if you get a very small, tiny, space, and the membrane cannot, restructure and reform, it will easily stick and possibly stop moving, and that would cause destruction. And so the red cell membrane skeleton is very important, especially when you look at these various proteins. So when it comes to categorization of these proteins, we have cytoskeletal proteins, anchoring proteins, and integral proteins. And so if you look at the integral proteins, I think we've already mentioned them here. We've also mentioned the anchoring proteins and cytoskeletal proteins. Okay, so a defect in any of these proteins can weaken and or destabilize the cytoskeletal structure of the membrane. And so you lose on the surface area, and you reduce the lifespan of the cell. And so this actually leads to early destruction of the cell. And of course, we know that hemolysis is early destruction of red cells. So any defect associated with any of these proteins will cause early destruction, and so hemolysis will easily set in. What are some of the membrane-related disorders that we can talk about? The first is congenital hereditary spherocytosis, which majority of the times is autosomal dominant. So you just need one person to pass it on to the rest of the people. But there's also a 25% recessive outcome. So it's never 100% dominant. You can also get a smaller percentage where you need both doses of the genes for it to manifest. It has an equal male to female ratio. And so males as much as females are affected. We've given different percentages in terms of the proteins that have been associated with congenital hereditary spherocytosis. And so when you look at spherocytosis as an outcome, as I did mention, spherocytes are small red cells with no area of central pallor. And so if you are to look at the causes of this kind of outcome, ankyrin would carry 50 to 60%, and then the rest of the proteins may carry other percentages. What do these cells go through? Basically, they have a reduction in the cell surface area. And, this basically increases their splenic clearance with chronic hemolysis. So when you have these cells with these defects, the cell surface area gets reduced. And so they get earmarked for destruction. It is the most common inherited hemolytic anemia in Europe and North America. This is not our bigger headache. Those of us who are excited to travel to the Americas, please watch even as you land there, because should you find a partner there, you could easily have congenital hereditary spherocytosis. Now, in terms of the clinical presentation and spectrum, there is a variation from mild, moderate, and severe cases, and of course, majority of the cases with this congenital hereditary spherocytosis tend to present moderately. Majority are actually moderate presenters with hemolysis. And there can also be hemolytic crisis, but the hemolytic crisis is precipitated more often than not by other underlying causes like infections. And so when you have an infective agent, because of this disorder, you would have severe anemia. Deep jaundice and subsequently splenomegaly would be key. And so if you find patients in certain regions presenting this way, if it is not in our setup here, you would tend to confuse this with possibly a sickler, because sickle cell anemia also has almost similar presentation where you have severe anemia, deep jaundice, and sometimes splenomegaly or gallstones. And you notice that gallstones is a mega presentation in this particular group of patients. But again, you'll see this also in sickle cell with aplastic crisis. And the aplastic crisis has also been associated and complicated by parvovirus 19. So these are patients, ladies and gentlemen, that we have to be keen and careful about because they would tend to present with symptoms that can easily be confused with other disorders. Malaria, for example, in our setup here would have similar presentations. And so one has to be keen on distinguishing this from a membrane disorder. The other types of membrane disorders include hereditary elliptocytosis. Hereditary elliptocytosis is autosomal dominant, affecting 1 in every 5,000, especially in the Caucasian communities, and then 1 in every 100 in certain African countries. Ladies and gentlemen, again, how many of us have hereditary elliptocytosis? 1 in 100 is a very large number. I think the whole continent should be exhibiting hereditary elliptocytosis at that rate. 1 in every 100 is a big, big, big number. I don't know how many we would be having. I think we would be having 5 million Kenyans with possibly, with hereditary elliptocytosis at this rate. But it presents with mild anemia in 90% of the patients. And so even if we have this particular disorder in our setup, again, rarely would we present with severe anemia, because majority of the patients have very mild forms of anemia. What about hereditary poikilocytosis? And these are patients where low heat causes some level of instability. So you have a decreased membrane heat stability that leads to severe forms of erythrocytosis. And this can be seen as early as in the neonatal stage of life. And we've seen a lot of patients in the Southeast Asian region presenting with ovalocytosis, and then you can also get what we call hereditary stomatocytosis. All these are membrane-related disorders that we can consider mentioning in our various setups. I think of importance for us is to remember that these disorders exist, and patients will tend to present with other clinical symptoms that may not easily be distinguishable from other causes of hemolysis. So if you are to look at the clinical features generally of any hemolytic picture of any patient, regardless, including membrane-related disorders, the first obviously is anemia, which might be acute or chronic. You would see jaundice in these patients. You would see bossing of the skull, especially if you are dealing with a chronic hemolytic picture. And bossing has always been associated heavily with the sickle cell. And so in our setup here, you must be careful about sickle cell. You will get signs and symptoms of any underlying condition. If there is an exposure to something, you'll see it. And then, of course, if you started investigating, you'll find dark urine, or someone has a crisis, this which might be in the abdomen, involving any of the organs, or you have renal failure, you have stones. So all these complications would easily come, in with some form of clinical symptomatology. So I think as we prepare towards a wind-up, it is important for us to very quickly mention some of the laboratory investigations that may be necessary. Obviously, for us to establish anemia, we have to take blood and try to, determine the type and severity of anemia. So the HB, the hematocrit, the total RBCs as part of the total blood count is essential. And as basic as it may sound, you must investigate this patient by doing a full blood count or a total blood count. The other thing that is of importance, obviously, is that you want to know the type of anemia. For most of this, we can also classify anemia based on morphology or size. If you have an MCV, for these patients, the MCV tends to be normal. So you would see a normochromic or normocytic kind of picture. But you can get a raised MCV, or a low MCV. But the low MCV then would have to be investigated further. A lot of times, you may see a normal picture. If you see an increased MCV, ladies and gentlemen, try to associate that possibly with the reticulocytes, because you'd have to have higher MCVs, especially as the cells increase in size. Okay, then you would question yourself, if the MCV is affected, what kind of anemia are you dealing with? Are you likely dealing with an anemia due to hemolysis or not? If the anemia is due to hemolysis, the key thing you want to establish is the reticulocyte count. Remember, I did mention that reticulocyte counts may be elevated in these patients. You would see what we call polychromasia. Polychromasia is basically nothing other than an increase or differences in terms of color. More cells would have different colors, and so the color change is evident. Poikilocytes are different shapes, and And so you can also see nucleated red blood cells. Other investigations may require that you check on the bilirubin levels and the lactate dehydrogenase levels. So all the things that I did mention when we started the lecture would also be affected. Okay. The other thing is you want to ask yourself the site of hemolysis. Is it intravascular or extravascular? If it is extravascular, you know you tend to see a splenomegaly, because then the reticuloendothelial system gets involved. For the other causes, it could also be intravascular, and we know that some of the enzyme disorders could also cause intravascular hemolysis, especially for G6PD when it causes an acute hemolytic picture. Okay, and so you can look at other laboratory parameters that may be of necessity at that point. You also may want to look at laboratory aspects to establish the cause. What is the etiology of the hemolysis? And so this forces you to look at the history. Supposing a patient told you to travel, I may have been exposed to malaria, and this may have a, I took antimalarials, and when I took antimalarials, this is what transpired. While you may think the hemolysis is associated with malaria, it may just potentially be the antimalarials that are causing hemolysis. And then, of course, the clinical features of hemolysis would be evident, and that would help you establish the cause. Then finally, you may want to look at specific investigations where you do a peripheral blood film, and the peripheral blood film would look at various characteristics of the cell. You may see blister cells or bite cells, and these are cells that look like they're eaten, they are bitten by the side. And so you see dents on the red cell. You don't see a uniform red cell, and that has been seen in G6PD. You'll also see these cells that we describe as spherocytes or ovalocytes or elliptocytes. You'll also see fragments, red cell fragments. We call them schistocytes, okay? And then you may see things we call Haynes bodies or other parts may be found within the cell. Then you may want to screen using different tests. Nitroblue tetrazolium has been tried for G6PD or fluorescent spot test, osmotic fragility, to look at other membrane-related disorders in general. And I think osmotic fragility is something that you may want to consider because it is also done for sickle cell, and it is good to look at patterns that you may see, may be unique for the two disease entities. You can also do a quantification assay for the enzyme that is of interest, and that might assist you to establish the quantities that may be missing and classify your patient appropriately. Sometimes we can analyze using different mechanisms, the membrane enzymes. Remember, we talked of electrophoresis, but you can also use other immunoassays to look at the various types of proteins, and that can tell you which of the proteins are affected. Last but not least, under specific investigations, you can also do molecular studies and look at the damage in terms of the chromosomes that are affected, and then you can do flow cytometry, and you can do an immune panel to exclude other causes of hemolysis. Ladies and gentlemen, we have a few minutes before 8, and I want to stop at that just to hear if there are comments or questions from the team, and then possibly we can answer as we close the day. Let me see if there are any issues in the chat box. We are very tired, sir. Okay, fine. I think I believe and agree with you. We are all very tired, and thank you for for writing this message that you are tired. Sorry for that. I hope you are not tired because the lecture was boring. And if it was, you'd have to bear with me. This is the best we can do for now. Okay. So let me not spend so much time because people are already too tired with the Coombs test. Let me hope that when you come to the department, we shall be able to explain the Coombs test. But basically, the main principle behind the Coombs test is to look for specific either epitopes or to look for antibodies on the cell surface using other antibodies. So you bring in an antibody. You get a cell upon which a protein is targeted by a specific antibody. So if you have an antibody that targets a specific protein on the cell surface, you will have that antibody binding onto that receptor or on that epitope. And so for you to determine that there is actually an antibody that has bound, you use an anti-antibody. So you get an antibody that targets that particular antibody. And that is what Coombs test is all about. We use antibodies to target antibodies towards the red cell. Okay, I hope that helps. But when you come to the department, we shall take more time to explain to you exactly how this is done, so that then you can tell that there is an antibody that targets a specific portion of the red cell. And so, as I did mention, you have immune and non-immune mediated types of hemolysis. Okay. I hope that helps. Any other comment? And if there isn't, ladies and gentlemen, let me respect the fact that you may be very tired, and I wish you a happy new year.